This is the validation study for the tip dance versus the non-tip dance protocol on the Easy One robot by Monia and Brittany Gibbs. The Easy One robot is an automated system used to extract DNA from forensic evidence samples. It contains two settings, the tip dance setting and the non-tip dance setting. On the left is a picture of the Easy One robot. It has a keypad where you can select which protocol you'd like to use. On the right is a picture of the screen where you would select which protocol you would like to use. Number 1 says trace, which is extraction without the tip dance protocol, and number 2 says trace TD, which is extraction with the tip dance protocol. When using the tip dance protocol, the filter tip moves back and forth while pipetting the samples so that it can pipette the liquid without pipetting any of the solid material. So you don't have to remove this material before putting your samples in the Easy One robot. When you do not use the tip dance protocol, the filter tip does not move back and forth, so you have to take the extra step of removing the solid material with forceps in a centrifuge spin basket before putting your tubes in the Easy One robot. Normally, we take the extra step and we do not use the tip dance protocol. The goal of this validation study was to determine the effects of using the tip dance setting on the Easy One robot using buccal swabs. 4 buccal swabs were obtained from 2 volunteers. 2 swab samples from each volunteer were extracted using the tip dance protocol, and 2 swab samples from each volunteer were used using the non-tip dance protocol. So that's 4 samples extracted using the tip dance protocol and 4 samples extracted not using the tip dance protocol. We would have liked to run more samples, however, due to limited resources, we were unable to run more samples and we were limited to a total of 8 samples. Samples were prepared using gill buffer, proteinase K, and DTT. These reagents are used for cell lysis and protein degradation. All the samples were extracted using the Easy One robot. Instrumentation was carried out according to the manufacturer's protocol. Quantitation of all the samples was carried out using the Quantifiler Human DNA Kit on the ABI Prism 7000 instrument. Reagents included in this kit are Tris buffer, Taq polymerase, primes, and TaqMan probe. Real-time PCR involves measuring fluorescent dyes in the samples as the DNA polymerase cleaves the reporter dye from the TaqMan probe. And on the left lower corner is a picture of the ABI Prism 7000. These are our results. As you can see, many of our samples had a very small amount of DNA. We believe that this is due to the presence of inhibitors. So to rectify this problem, we ran our quantitation over again. This is our second plate of quantitation. As you can see, we got better results and a higher quantity of DNA in each one of our samples. Overall, the total average of the DNA in the samples extracted without the tip dance setting was higher than the amount of DNA found in the samples extracted with the tip dance settings. This is likely due to sample number 6, which has a higher amount of DNA than all the other samples. This is likely an outlier. However, we would better be able to determine how far off these results are if we were able to run more samples. But as I previously explained, due to limited resources, we were not able to do so. Outside of sample number 6, the other quantities are similar. For example, sample number 1, which was a sample extracted with tip dance setting, and sample number 8, which was extracted without the tip dance setting, have a similar quantity. All the samples were amplified using PowerPlex 16 Hot Start Amplification Kit on the 9700 thermocycler. Master Mix Primer and 9947A were used in the kit. 9947A is the human DNA found in this kit. Our target was 2 microliters of DNA for each sample. On the lower left-hand corner is a picture of the 9700 thermocycler. Sorting of the amplified DNA fragments of the samples was carried out using the ABI 3130 genetic analyzer. The results were shown using the GeneMapper ID software. This is a picture of the genetic analyzer. Results revealed that there is not much of a difference in the use of either method. As explained before, the quantities were similar outside of the outlier. There was a small amount of contamination present in the results for both methods. This is likely due to an error made by the technician. The contamination was seen as small peaks on the electropherogram that weren't any of the alleles that were called. Neither method made much of a difference in the amount of DNA yielded or in the amount of contamination. Therefore, it is our recommendation that the tip dance protocol be used at the technician's discretion. On behalf of Monia Bailey and Brittany Gibbs, I thank you for your time.